Today we're going to move on to topic 9. This has to do with DNA, RNA, and protein. We can break these big ideas into the structure of genetic material itself, the way DNA replicates, the flow of genetic information from DNA to RNA to protein, and sometimes I talk about genetics of viruses and bacteria. I normally don't spend a lot of time on this. That's largely because it's so variable. For example, viral genomes can be DNA-based, RNA-based, linear, circular, single-stranded, or double-stranded, and this makes for a lot of information to go through. Each type of virus is going to express its genes and replicate slightly differently. However, in light of the fact that we're dealing with COVID-19, I will talk about this a little bit in an extra video that I'll be posting. If you're curious, COVID-19 is a linear RNA-based virus. But for today, we're going to focus on how your DNA works, how it's going to replicate, and how you express genes. So we're going to start by thinking about the structure of the genetic material itself. So remember that DNA and RNA are polymers of nucleotides. That means that they consist of long chains of different monomers. Those monomers, being nucleotides, are then put together to make the polymer itself. Remember that a nucleotide is made up of 3 major parts: a nitrogenous base, a sugar, and a phosphate group. The possible nitrogenous bases that we can use are going to be one of 2 types. Either they will be a pyrimidine, such as thymine and cytosine, or a purine, such as adenine and guanine. Notice that pyrimidines only have 1 circle of carbon, whereas purines have 2. And they're always going to pair pyrimidine to purine. One of the things that we want to keep in mind when we're thinking about how DNA works is the structure of the nucleotide. When we're thinking about carbons, we're going to count them. So there are 5 carbons that are involved. The first carbon, carbon number 1, is going to be located where the base binds to the circle. Then we count carbon 2, carbon 3, carbon 4, and carbon 5. So the phosphate group is always going to be on carbon 5. Now we have to care about this because carbon 5 and carbon 3 are going to be very important for building DNA polymers, and knowing where they are is going to help you understand how replication happens. Each of the strands of DNA itself is a polynucleotide, which is a polymer, and it's going to be bound sugar phosphate, sugar phosphate. You can see in the picture here that the sugar itself is going to alternate with the phosphate. This is going to form a covalent bond that joins the nucleotides together to make the polymer. That means that we can actually give names to different ends of the DNA. So the end of the DNA that ends in a phosphate is going to be called the 5' end, and the end of the DNA that ends in a sugar is going to be called the 3' end. Now DNA itself is made up of multiple polynucleotides, right? Because it's double-stranded. So the way that this works is that there's hydrogen bonds in between the pairs that hold these strands together. So for example, A and T are going to pair together. The hydrogen bonds between A and T will hold the polynucleotides together. C and G will pair together. The hydrogen bonds will hold those together. So this is what actually keeps DNA as a double-stranded helix. It's important to note here that these strands are reversed from each other. So the strand on the left has the 5' end, the phosphate end at the top, and the 3' end at the bottom, whereas the strand on the right has the 5' phosphate end at the bottom and the 3' end at the top. Right? So these things are in the opposite orientation, and they have to be in order to fit together. So the hydrogen bonds in this opposite orientation are going to allow the DNA to form this double helix-based structure. We can illustrate that structure in a variety of ways. So for example, we have a ribbon model on the left, the partial chemical structure I showed you before in the middle, and a computer space generating model on the right. All of these images mean the same thing. There's always going to be a sugar phosphate backbone that's going to alternate, and there's always going to be pairs of nucleotide bases in the middle. The full name of DNA is deoxyribonucleic acid, with nucleic referring to DNA's location in the nuclei of the eukaryotic cell itself. RNA itself, ribonucleic acid, is unlike DNA in that it uses a different sugar. So that sugar is going to be ribose instead of deoxyribose. It also has a different nitrogenous base, that's going to be uracil, and that will replace thymine. Otherwise, their structures are extremely similar. Now, if you're still unfamiliar or uncomfortable with the structure of DNA and how it fits together, there's a lovely video for you inside the PowerPoint presentation that's been posted, or you can go back to our original topic on how nucleotides and polynucleotides work way back in Chapter 3. The discovery of DNA was really contributed to by 4 different people. Maurice Wilkins was the first person to start looking at DNA and how DNA could be structured. Rosalind Franklin determined that the DNA structure had to be helical in nature, and Watson and Crick really took the information that Maurice and Rosalind put together and built the physical structure. In 1962, they were all awarded the Nobel Prize. Well, at least Watson, Crick, and Maurice were. Rosalind would have received the Nobel Prize, except she died from cancer in 1958. And Nobel Prizes, unfortunately, are never awarded posthumously. The important thing here is that the Watson-Crick model gave a new meaning to the words genes and chromosomes and gave us the information that genetic information in a chromosome is encoded in the sequence of DNA. That's because sequence is the major difference between genes. The structure of the DNA itself is not very different. Now we're going to think about how DNA is able to replicate itself. And that's really important because it's necessary for cells to go through division, and that means it's necessary for us to reproduce. Remember that DNA replication occurs during S phase of interphase, and this needs to happen to let each daughter cell have a copy of every chromosome. There are 3 hypotheses about how DNA replication could occur. The first of these is called the semi-conservative model. In this model, the strands of DNA separate and each one is used as a template to synthesize the new strand. In the conservative model, the entire original DNA stays together but is used to create an entirely new molecule of DNA. And then a dispersive model, different parts of the DNA are used to synthesize other parts, creating no original parent copies of DNA. It's now known that DNA replicates through the semi-conservative model. This means that the 2 DNA strands separate. Each strand then becomes a template for the assembly of the complementary strand, and each new DNA helix has 1 strand with old DNA and 1 strand made of new DNA. In order for DNA replication to occur, first an enzyme has to unwind the DNA double helix. This is possible because the helix is only held together by hydrogen bonds between the base pairs. A different enzyme is going to attach complementary DNA nucleotides to each original half, and this will result in 2 identical sister chromatids. So essentially what we're doing is we're using the template of the original DNA and the fact that nucleotide bases are only going to pair with 1 other base to make a perfect copy of the other side. Now we'll watch a video explaining this and then we'll unpack it a little bit.
In principle, copying DNA, a process called DNA replication, is very simple. The 2 complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding 2 identical DNA molecules.
There are 3 enzymes that we're going to need in order to do what we saw in the video. The first one is DNA helicase. This is the enzyme that will unzip the DNA so that other enzymes can have access to the DNA strands. Using the enzyme DNA polymerase, the cell will then synthesize one daughter strand as a continuous piece. DNA polymerase can only add new bases to the 3' end, so this is the end that's going to grow. The other strand is synthesized as a series of short pieces, which are then connected by the enzyme DNA ligase. So helicase opens, polymerase extends, and ligase connects. Remember that the strands of DNA are different from one another, and we can think of them based on what the end consists of. Ends that have phosphates are 5' ends, and ends that do not are 3' ends. The replication of DNA can only occur from the 5' to 3' direction, because DNA polymerase can only add new nucleotides to the 3' end of a growing DNA molecule. This means that there is a direction that the DNA is going to grow in. If we look at the replication fork here, we can see that on the top strand, this DNA polymerase molecule can synthesize DNA in one piece, because as the replication fork opens, it's still going to be moving in the 3' direction. However, on the bottom strand, that's not the case. As the replication fork opens, a new DNA polymerase will have to join in order to synthesize a new strand. We call the strand that synthesizes one piece the leading strand, and the strand that is synthesized as multiple pieces the lagging strand. And these special pieces have a name, Okazaki fragments. In the end, DNA ligase will connect all the Okazaki fragments together to make one piece of DNA. In the next piece of the big picture, we'll be talking about how we get from DNA to RNA to protein. The DNA of a gene is a linear sequence of many nucleotides. It's going to need to be transcribed into RNA, and that will eventually be translated into a protein. So how does all of this happen? Different regions of chromosomes have different genes. And genes are sequences of DNA that encode a protein or a function. Different nitrogenous bases on the same region of homologous chromosomes can lead to different alleles. So for example, in the right here, we have a small difference in this gene. In one version, there's a CG pair, and in the other version, there's a TA pair. This would be an example of different alleles. One of these alleles might encode one color of eyes, like blue, and the other might encode another, like brown. If we want to express the eye color that these encode, we would need to transcribe the DNA into an RNA. This is called transcription because it is like making a copy or a transcript of something. Then we would want to convert this into the protein that it represents. This would be translation. In this case, we are moving from the nucleotide sequence of RNA to the amino acid sequence of a protein. It's like translating a language. In order to make a transcript, DNA helix has to be unzipped. RNA nucleotides have to line up, and the RNA polymerase has to join them along the strand of the DNA, following the base pair rule. RNA transcription has 3 steps: initiation, elongation, and termination. So let's talk about what is included in each of these steps. In order for RNA synthesis to begin, an RNA polymerase must find something called a promoter. A promoter is a region of DNA that tells the RNA polymerase that a gene starts here. It also gives information about where the gene should be expressed. We'll talk a lot more about promoters and other regulatory regions when we talk about how genes are regulated. Once the RNA polymerase has bound to the promoter, initiation can move forward. The next step is elongation. During elongation, RNA polymerase will read the DNA and will add free RNA nucleotides to make the RNA. This will continue until the RNA polymerase reaches the termination sequence. The terminator DNA or termination sequence tells the RNA polymerase that it has reached the end of the gene, and therefore the RNA is complete. This is the termination step. If you're not comfortable with how this works, there's a lovely animation about it in the PowerPoint. Once an mRNA is produced, it's not quite complete. mRNAs contain multiple regions of information. These regions are often called introns and exons. Introns are regions of genes that do not code for any protein. And exons are the regions of genes that have the instructions to make a protein. Most of our DNA does not actually code for proteins, and most human genes have multiple introns in them. This means that there's information inside the mRNA that we don't need for translation. In addition to that, there is a cap and a tail, and these are important for helping the mRNA stay stable inside the cell and allow it to interact with the ribosome that's going to translate it. Before we can go through translation, we are going to need to cut out the introns, the regions of the gene that don't encode for the protein. And then we are going to use enzymes to join the ends together to create an mRNA that includes a cap, a tail, and only the part of the gene that actually encodes the protein that we want to translate. We'll talk a little more about why we have a process like that when we get to gene regulation. Once we have this mature mRNA, then we're going to have it leave the nucleus and enter either the cytoplasm or the endoplasmic reticulum, depending on where it needs to be translated. The kind of RNA that encodes amino acid is called a messenger RNA, or mRNA. It's called that because it contains a genetic message that's going to be used by the translation machinery. Translation requires converting nucleic acid language into polypeptide language. So for example, if we have a situation where we have the following DNA, AAA, CC, GGC, quadruple A, we would want to create a transcript of that. That's going to be the complementary sequence. This complementary sequence can be divided into what we call codons. Codons are non-overlapping three-base words. Those words form a sentence, and that sentence can be used to add amino acids. So for example, UUU is a codon that encodes square here, whereas GGC encodes the circle and CGU encodes the triangle. Each of these is a different word, and it corresponds to a specific amino acid. The genetic code is a set of rules that dictates the amino acid translations of each mRNA nucleotide triplet. We can think about this in two different ways. On the left is a key that uses a circle. In order to read this, you would start in the center. For example, when we thought about UUU above, we would start and then move up and to the right into the U, then move up again into U, and then move up again into U, telling us that the amino acid sequence is phenylalanine. We can use the same system on the square key on the right. In this case, we would go to the top row, that's U, the first column, that's also U, the third U would also tell us Phe, or phenylalanine. It's a good idea to practice using a key like this, and there's a few different example problems that you can solve posted on Canvas. Now that we kind of understand how to look at the rules, let's talk about who does the decoding. So the translation process is done by tRNAs or transfer RNAs. These are RNAs that read the codon in the mRNA and help attach the appropriate amino acid. There are a lot of different ways to look at tRNAs. So for example, you might see them drawn on the right as a basic chemical diagram with a sequence. You might see them as a ribbon diagram, or you might see them as this kind of lumpy cartoon, but they all mean the same thing. There's a location called the anticodon, which is going to pair with the codon in the mRNA, and there's an amino acid attachment site up at the top of the tRNA where the appropriate amino acid is going to be attached. In addition to that, the tRNA itself is going to interact with an enzyme. That enzyme is going to catalyze the bond that will happen between amino acids. So what does this process look like? Well, an mRNA is going to go to the ribosome. A tRNA goes to the ribosome too. It's going to be looking for the start codon, which is always going to be the same, this AUG, or MET. When the start codon is found, it's going to add methionine. The next tRNA is going to read the next codon. It's going to go into the pocket, read the codon, and catalyze the reaction to bond amino acids together. And this process will continue until the new protein is formed. So we can look at it a slightly different way. So there are two sites inside the ribosome, the P site and the A site. tRNAs are going to enter into the A site. They're going to see whether or not they bind the codon with their anticodon. If they do, the enzyme will catalyze the amino acid reaction. Then, the tRNA that's located in the P site will leave, now that its amino acid is no longer attached. The tRNA that's in the A site will shift over to the P site, allowing room for a new tRNA to enter into the A site. And the cycle will continue until a stop codon is read. When we're looking at a chart like this, we can see that there are specific codons for start and stop. The AUG or MET or start codon is always the same, but stop codons can be one of 3 different sequences: UAA, UAG, or UGA. The rest of these are going to code for specific amino acids, and there are 20 amino acids in all. The reason that there are so many is so that there's redundancy. We'll talk about that when we talk about mutation. So based on how we talked about translation, we can see that it is also divided into the same 3 phases as transcription. Initiation, where the tRNA is going to read the start codon. Elongation, where tRNAs are going to come and match their codon, adding new amino acids to the sequence of the protein. And termination, when the stop codon is read and the tRNA ends the protein. Now we need to talk a little bit about mutations. Mutations are changes in the genetic information of a cell or virus. They're usually caused by errors in DNA replication or recombination, but sometimes they're caused by other things. A physical or chemical agent that causes a mutation is called a mutagen. Mutagens fall into several different categories. For example, radiation can be a mutagen, which is why sun exposure can cause cancer and why you have to be careful about X-rays. Chemicals can also be mutagens, like the carcinogens found in cigarettes, or preservatives in processed foods, and some cosmetics and cleaning products. And other options are infectious agents, like HPV and Heliobacter, which can also cause mutations. That's why viruses and bacteria are sometimes associated with cancer. Whenever DNA damage happens, which is the case for all 3 of these types of mutagens, or a replication mistake is made, the cell will try and repair itself. Whenever that repair process fails, that's when a mutation occurs. All mutations fall into 2 major categories. They're either synonymous or non-synonymous. Synonymous mutations change the codon sequence, but do not change the amino acid that it codes for. You can think of these like synonyms for words. For example, you might use rapid and fast to mean the same thing. Non-synonymous mutations change the amino acid that the codon encodes for, so these are words that do not substitute. Let's think about some examples. We're going to start with the sequence here, which represents normal hemoglobin. In the DNA, the sequence for glue is CTT, and the codon is going to be GAA. If we have a mutation that affects the second nucleotide and converts it to an A, this would change the codon to GUA. This results in a substitution of valine for glutamate. This particular mutation is common in sickle cell anemia. There are names for different types of mutations, but we'll only be talking about 3 of them today. The first is a base pair or nucleotide substitution, the second a deletion, and the third an insertion. We'll talk about what they are and how they will make changes to the DNA and the codons that are inside. The base pair substitution is a very straightforward one. In fact, that's the example we gave before. In this case, only one base pair or nucleotide has been changed. We've gone from GGC to AGC. This is a non-synonymous mutation converting glycine into serine. Notice that when a base pair substitution occurs, it only influences the codon in which it occurs. It will not have any effect on subsequent codons, for the most part. Deletions are pretty much what they sound like. One nucleotide or base pair has been removed. In this case, everything from the codon where the deletion occurred and downstream of it will be affected. So we go from UUU to UUG. But since everything has slid over to fill in that missing U, all of the downstream has changed. So instead of having Phi-Gly-ALA, we now have Lu-ALA. Mutations that affect all downstream nucleotides are called frame shifts. Effectively, they're shifting the frame that is being read by the translation machinery. This allows really small mutations, a single base pair deletion for example, to make huge changes. Now, can you think of a way that you could have a deletion that would only affect a single codon? This is possible if you delete the entire codon together. If instead of deleting one U, we deleted all 3 U's, Phi would be missing. So we'd go met, lice, gly, ala. It's possible for deletions to affect only one codon, but only when they are divisible by 3. Insertions are basically the opposite. Instead of removing a base pair nucleotide, we're going to add one. This is going to shift all subsequent base pairs downstream and affect all of the following amino acids. So instead of having phi gly ala, we now are going to have lu trip arg. So those are the three major types. We can either change an individual nucleotide, which should only affect the codon in which that nucleotide resides. We can delete a nucleotide, or we can insert a nucleotide. And both of these mutations are likely to affect everything downstream, unless they're divisible by 3. Now let's think about a problem. How could you have a single nucleotide substitution that results in a protein that is short, i.e., some of it is missing? If we go back to look at the genetic chart, we can see that there are a lot of different codons. Some of these codons are necessary for determining whether or not translation machinery should continue. The end of a protein is encoded by a stop codon, UAA, UAG, or UGA. If we had a situation where we converted something like UGU, which is cysteine, to UGA, we would create a premature stop codon. This would cause the protein to end early and make it short. Now that we've talked about how translation works and how mutations work, I want to ask you another question. Why do you think there are so many redundant codons? If we look at the codon chart, you can see that quite a few amino acids are encoded by more than 2 or 3. Leucine, or LEU, is encoded by 6 different codons, for example. So why are there so many? The first thing we should think about is, what are our options when we think about how big codons can really be? We have a limited vocabulary and a limited number of letters. So we only have 4 options, right? A, T, C, or G. How many words could we make if each word was 1, 2, or 3 letters long? We can calculate the total number of words by using the equation 4 to the n, where n is the number of letters in a word. 4 to the 1 would be 4 words possible if each one has only 1 letter, 4 squared if 2-letter words are possible, and 4 cubed if 3-letter words are possible. So we can make 4, 16, or 64 words respectively. How many words do we need? How many amino acids exist? There are 20 different amino acids. So if we only have 1-letter words or 2-letter words, we don't have enough to make one for each of the amino acids we need to make. So in order to have at least 20 choices, we need to have at least 3-letter words. This also means that we have a lot of extra or redundant codons that we can use to protect ourselves from mutation. So for example, if we look at leucine again here, we can see that if anything starts with CU, the third base in the codon doesn't really matter. This helps protect us and is where synonymous mutations come from. So not only does having 3-letter words give us enough words for the 20 amino acids we need to make, it also gives us extra codons that we can use to protect ourselves from random mutation. Alright, so let's see if we can use what we've learned so far to talk about how COVID-19 replicates. At the beginning of this lecture, I talked about how there are a lot of different ways that viruses can have their genomes. They might have a genome that's based on DNA or one that's based on RNA. It might be linear, circular, single-stranded, or double-stranded. Based on the way the virus is structured, that's going to determine how its DNA is replicated and whether or not it goes through transcription before translation occurs. The most simple one to think about is double-stranded DNA viruses. Examples of this are adenoviruses and herpes viruses. These are going to pretty much work the same way you do. They're going to have regular double-stranded DNA, and that DNA is going to be replicated using a DNA polymerase that's DNA dependent, just like yours, and a DNA dependent RNA polymerase to make mRNA, just like yours, and it'll go through translation the same way you do. So these viruses are really not that different from you or I. The next option is single-stranded DNA viruses, like the parvovirus. These are also pretty similar. In this case, we're going to have one piece of DNA that's going to make a complementary copy. Now we have double-stranded DNA again, so everything is going to work pretty much the way it works in you. When it gets to RNA viruses, that's where things get a little bit tricky. You don't have any enzymes that are able to read RNA viruses and make replications of them, or make complements. So in order for an RNA virus to do anything with its genome, it has to bring with it a special RNA-dependent polymerase. In the case of double-stranded RNA viruses, it will behave exactly the same as double-stranded DNA would, except the virus will use its own RNA-dependent RNA polymerase to do everything that the DNA polymerase would be doing normally, and that the RNA polymerase would do for transcription. Single-stranded RNA viruses are a little different from that. They're still going to need to bring with them an RNA-dependent polymerase, but it matters whether or not the RNA is the positive strand or the negative strand, because your cells are only able to read positive strand RNA and translate it properly. This means that positive strand RNA viruses are able to go ahead and make proteins directly from their genomes. They're going to use the RNA-dependent polymerase to make a complement, negative RNA, that they'll then use to make more of their positive strand RNA to send on in their offspring. Negative sense RNA viruses make messenger RNA directly from their negative strand RNA using their RNA-dependent polymerase. They also make positive strand RNA so that they can make more negative strand RNA to send off in their offspring. This means that they have to go through both transcription and translation, whereas the positive strand RNA virus can go directly to translation. There are a few other types of viruses like retroviruses and adenoviruses, but today we're going to be skipping those. So, which one of these is COVID-19? COVID-19 is a single-strand RNA virus, and it's positive strand. So this is essentially how it works. When the SARS-CoV-2 or COVID-19 virus comes into contact with your cells, it's going to bind to the receptor ACE2 on the cell surface. This is going to have the cell bring in the virus via receptor-mediated endocytosis. Once inside the cell, the virus will open and allow the RNA to enter into the cytoplasm. Some of that RNA will be used to directly translate viral proteins, and those proteins will be used to make a replication complex so that the RNA can replicate itself. Proteins and RNA that are made this way will then be assembled into a new virus in the Golgi and will eventually be released through exocytosis. So let's think about what is different and the same between you and the COVID-19 virus. Only one thing is actually the same, and that's the way translation works. So messenger RNAs, or single-stranded positive strand RNAs, are read by ribosomes and translated using the same codon chart. Everything else, on the other hand, is different. The viral genome is RNA, whereas yours is DNA. It's single-stranded, not double-stranded like yours. Single-stranded RNA genomes are directly translated, if they're positive strand, and no transcript is made first. And the RNA is replicated by proteins that are encoded in the viral genome, because it needs an RNA-dependent RNA polymerase. So as you can see, viruses like double-stranded DNA viruses are pretty similar to you, but viruses like COVID-19 are very different. It's important to understand how a virus replicates to try and interfere with how it might grow, and thus find a way to combat it.